My name is Joe Dickinson. I'm the president of the Ontario Forage Council, and welcome to tonight's um, tonight's first night of Profitable Pastures. First of all, thank you to our conference sponsors, Brussels Agri Services, Range Ward, and the Beef Farmers of Ontario. Um, we really can't do this without the support of our sponsors. Thank you also to tonight's speaker sponsor, that's Trillium Mutual Insurance. Tonight's presentation is Growing Grass, Supporting Livestock, Nitrogen and Pasture Systems with Caroline Reisiger. Caroline is a PhD candidate in the Department of Plant Agriculture at the University of Guelph, where she studies pasture-based livestock systems and forage management. Originally from a small Christmas tree farm in Brant County, she completed her BSc in 2020 in plant science and MSc in 2022 in molecular and cell biology at the U of G, and now focuses on improving grazing recommendations in Ontario through applied research on nitrogen fertility, forage utilization, and alternative forage species in perennial pastures in pursuit of her PhD. Her work includes field-scale grazing trials and on-farm measurement tools to help producers increase pasture productivity while considering economic and environmental outcomes.
Tonight, as Joe gave me a great introduction and said that I'm going to be talking about growing grass, supporting livestock, and nitrogen in pasture systems. So this is my main PhD project. I looked at nitrogen fertility in pasture, and I conducted a trial at the Elora Research Station in on pasture, and so we're going to get right into it. Okay. So, if you've heard me do a couple of talks before, any of my talks before, I kind of start by saying that sustainable livestock systems must be profitable. And we know that there are many benefits for having pastures in the landscape, but they won't stay there without the production and economic reasoning to back them up, or any of the other scientific knowledge that we need to gather to talk about pasture systems in the landscape. Tonight, we're talking about profitable pastures, and I'm hoping that some of this will have that focus. So, one way to increase profitability on pasture is to increase forage yield. And this improves land use efficiency. One way to do this is to potentially apply a nitrogen fertilizer. And this could boost forage yield and could improve land use efficiency, but there are also potential environmental concerns associated with nitrogen use that we have to keep in mind, and some of these we have actually looked at or are going to look at in this project. So currently, we don't have any pasture-specific nitrogen recommendations in Ontario, and this project won't give those. We are also lacking information on legume persistence with nitrogen application in Ontario pastures, and we also need production data, both plant and livestock production, to bring context to nitrogen use on pasture. And these are two things that the project that I'm about to talk to you about will focus on. We are also looking at some other aspects of nitrogen fertility on pasture, including root biomass and carbon sequestration. So, this research was conducted on a 64-acre pasture trial in Elora at the Elora Research Station, and it was 3 years long. And it had four different experimental rates of a polymer-coated urea. This is a slow release fertilizer that were applied in each spring before green up. And the rates were 0, 30, 60, and 90 kilograms of nitrogen per hectare. The pasture itself was divided into 8 rotationally grazed fields. And I'm going to show you a map in a second to describe this a little bit in a little bit more detail. But the goal of our rotational grazing system in these pastures was to maintain the same exit residual on all of the treatments. And we stocked the fields accordingly. So instead of potentially having nitrogen treatments where there was a lot more biomass produced that wasn't being used by the animals that were in there, or potentially extending the grazing period or changing that at all, we decided to alter the stocking rates. So the trial was, this is a satellite image of the trial, and we split it into two blocks. Each block had our nitrogen treatments in them. So there was one set of four and another set of four. And we had um, each one of these fields is 8 acres. So on each 8 acre field, we had one group of cattle that was rotating through the paddocks on this 8 acre field. And these fields were split into 8 paddocks. So each one of these paddocks was 1 acre, and the animals would rotate through the top four and then into the bottom four. We only did measurements on the top four, so 1, 2, 7, and 8, and but the nitrogen treatment was placed on all 8 paddocks in this field. And the way that we split up the paddocks and the way that we split up the nitrogen treatments and the way we did our rotations, which I'm going to talk about later in this presentation, were conducted in this way because of the experimental design, our need for replication, and how we wanted to be able to evaluate the nitrogen effect. We also had an additional plot scale trial where we tested different components of the mix of our pasture. Now is a good time to tell you what exactly was the mix on our pasture. So it was a cool season perennial mix. We had legumes that were red clover, white clover, alfalfa, and trefoil. And we also had a mix of grasses, which included orchard grass, tall fescue, meadow fescue, Kentucky bluegrass, and perennial ryegrass. And those were all mixed together on our pasture, but on our plots, we split those all into different plot strips. We also tested two different cutting heights, and the nitrogen rates were the same as on our pastures. But today, this talk is for profitable pastures and not profitable plots. So I'm just going to be presenting the pasture mix data today to help support the pasture data that I have. So first, I'm going to tell you what our project has found so far in terms of results, and then I'm going to show you some of the graphs and data to back this up. So what we found is that there were limited forage quality changes between nitrogen treatments. We also found that applying nitrogen led to higher forage yields. This makes sense. We also found that the increased nitrogen led to increased stocking rates and a greater overall net calf production in those nitrogen treatments. And we found that these yield or biomass increases were primarily due to the increase in grass growth, the increase in grass biomass, and not the increase or decrease in legume or weed biomass. And we also found similar results on our complementary plot trial. So, first thing we're going to do is get into some forage quality data. I said there was limited forage quality changes. One change we did measure and we were able to find was a difference in crude protein amount in the different nitrogen treatments. So, for example, the 90 kilogram per hectare nitrogen had more crude protein in the forage samples than the 0 kilogram per hectare. This difference was only observed in May and not in August. And if you can remember, the nitrogen treatments were applied before green up, so this is end of April. And so the August sampling was quite far away from any nitrogen application that we had done. We also did do some limited sampling in June, and there was also no effect on crude protein by June. So the crude protein effect that we are seeing here and that it's graphed here is a temporary one, and it is one that we see probably in May exclusively. And there were limited forage quality changes, so I wanted to highlight some other forage quality metrics that we analyzed and we looked at statistically, but had no effect, or the nitrogen had no effect on them. So these include some digestibility metrics, but they also included some energy metrics. So even though the forage quality data resulted in, or we saw crude protein increases, we did not also see higher energy amounts in in any treatment. If you look down each one of these table rows or table columns, you'll see that the numbers are fairly similar, 68, 69, 70. They are very similar across each one of the nitrogen treatments. So now we're going to turn a little bit to forage biomass. So this is a graph of consumed biomass. So we measured the amount that the livestock ate, basically by the amount that was missing after they left. And it's graphed by nitrogen treatment. So each one of these colors is a different nitrogen treatment, and it's split into different years. The first thing I wanted to talk about here is the variability between years. In 2023, Elora had a very wet year. And over the whole season, we actually had 545 millimeters of rain. And the other 2 years, 2024 and 2025, were a bit more typical, with 2025 actually being a little bit on the dry side, especially at the end of the year. We had 100 millimeters less rain in 2025 than we had in 2023. And you can see that in actually the amount of forage that was available for the animals to eat. There's a similar trend when I look at, or when we look at cumulative biomass over the season compared to this consumed biomass that I have on the graph. The next thing that I wanted to talk about was the differences in yield, or differences in the biomass that was consumed. If we're looking at this graph, there is a nitrogen effect. So there was more biomass being consumed in the 90, for example, than there was in the 0. And this is also true in terms of yield, which I will show in a little bit. So the next thing that I wanted to talk about was that livestock rates, we we altered them. So there was more livestock on the higher nitrogen treatments than on the 0. There was actually more livestock on the 30 kilogram per hectare in most years. In 2024, it was actually very, very similar, based on forage availability. So we consulted with our farm manager at the Elora Research Station. And what we didn't want to do was underfeed the cattle. Our plan was to try to put the number of animals that that pasture could feed on that pasture so that all of our calves were able to gain the same amount of weight. This trial had cow-calf pairs on it. So a important piece of information is that we were overall successful. The amount of calf gain on each nitrogen treatment was statistically the same from entry to exit. I haven't spliced out that data over the year. We do have a few interseason measurements, but from entry to exit, the calves gained the same whether they were on the 0 nitrogen treatment or on the 90 nitrogen treatment. What did change was actually the amount of animal unit months per acre. So because we had more cattle, more cow-calf pairs on the higher nitrogen treatments, we were able to get more animal months per acre. And this was the case in all 3 years. So, to translate animal months per acre to actual numbers of pairs that might be something that is you can conceptualize or see the number of animals on our pasture. These were the average number of animal pairs on each one of the fields. So each 8 acre field. Now they are obviously, we didn't have, zero point 0.6 of a cow-calf pair. This is an average across all 3 years. So the number of animals would have varied in 2023, 2024, and 2025 in order to make that 7.6 average. We did see this stocking rate change in all years. We were able to have the pasture support more animals and if it was a higher nitrogen treatment. And I also wanted to mention that this is overlaid on a average cumulative biomass in a dry matter kilogram per hectare over all 3 years. So this would be the average biomass accumulated each year by the pasture. Now, we found that for every 1 kilogram per hectare of nitrogen applied, there was 11 kilograms per hectare extra forage biomass produced. And because of this extra biomass produced, we were able to put extra animals on that pasture. An interesting aspect of my research was also in the amount of pre-grazing biomass that was there in each rotation, depending on the year. And on our pastures, we had 7 rotations, which I totally understand is a ton of rotations. That's a very quick rotation speed, particularly for Ontario. But recall back to the way we had set up our fields with those 8 fields and the 8 acres per field and the 8 paddocks per field. So each group of animals only had 8 possible paddocks to move through. And because of this, we ended up with 7 rotations. Now, this graph is showing the pre-grazing biomass and in each rotation, and it's split by year. So in 2023, it was a wet year, but we had a dry spring. So it was dry, pretty dry in rotation 1 and 2. So this would be May, June, and then by the end of June, we'd actually had some rain. So this resulted in a pretty steep increase in forage biomass pretty quickly, a late spring flush. And then because it continued to rain for the majority of the rest of the year, this rotation um, amount or the amount per rotation stayed relatively stable. Now, in 2024, 2025, we had more typical years where we had a spring flush and more of a summer slump into the season. Now, the 7th rotation was at maximum mid-September when these animals were removed off of the pasture. So this graph is going from the beginning of May to the beginning of September. Another way of looking at this is splitting the pre-grazing forage harvests by rotation and by nitrogen treatment. So an interesting part about this graph is that the nitrogen effect that we were seeing on our pastures existed for the first 3 rotations, but did not continue to exist after the 3rd rotation. The 3rd rotation ended about the end of July or end of June, beginning of July. So the 4th rotation is starting at the beginning of July. Now, we did a single application of nitrogen for these pastures in a slow release form. The slow release form was a 60-day slow release. So this is showing that there is an effect for about 60 days from the end of April until the end of June or the beginning of July, about 60 days of nitrogen effect. So that's an important part of our research and something to keep in mind that we were seeing the nitrogen effect when the nitrogen presumably was being released, and then we stopped seeing the nitrogen effect after that nitrogen was released. Now, another thing I wanted to get into was a plant type and cumulative biomass. So this here is a graph of the biomass and nitrogen treatments. And this time, the biomass, the total cumulative biomass in each nitrogen treatment, and it's split by year. So we have 2023, 2024, and 2025. And here is the key over here. So the grass is green, the legumes are blue, and the weeds are red. And what we found was that there was an increase in grass biomass, but there was no significance in the legume biomass. So the legume biomass in each individual year did not change. Now, because these pastures were treated repeatedly with the same nitrogen treatment across the 3 years, what you'd expect if you were going to be observing a legume persistence problem that's occurring within the first 3 years of this nitrogen treatment is you might see that legume biomass start to or move towards being less by the 3rd year. But these differences weren't significant. In 2023, there is what looks like, a pattern, but the error was very high for our 2023 year. And this is part of the reason why these values are not significant. So, what we're observing here is that the significant change in forage biomass is because of the grass biomass increasing. Another important thing about our pastures was that because we had these 7 rotations, we kept the pastures relatively short. We're not letting the grasses really grow to their full height for really any time during the year. And this is really important, and we believe this is really important for why we're seeing this effect where the legumes are staying in the pasture. And this is because legumes and grasses compete for light. And when the grasses receive nitrogen, they are better at competing for light. But grasses are also better at competing for light in general because the grasses can grow taller than the legumes, so they can shade out the legumes. So by keeping the pasture short, the grasses, even though they had a lot of extra nitrogen on the higher nitrogen treatments to be able to grow, they weren't able to shade out the legumes and therefore outcompete them. So that is an important piece of information for our trial and probably why we're seeing what we're seeing. One caveat to this is that the actual percentage of legume that we were seeing in our samples we were pulling for composition, so our composition samples to be able to make this graph to figure out what was in our pasture. After harvesting with our haul drip forage harvester, certain sections of the pasture to get an average biomass in the pasture, we took some forage from that pile that was removed by the haul drip and we put it in a bag and then we went back and we sorted it at the station, sorted it into its different components. And in those bags, the legume percent composition did decrease in the different nitrogen treatments. However, because the biomass also increased in the nitrogen treatments, we found that this did not translate to a biomass decrease because the biomass was staying relatively the same, but the percentage of grass in the samples was increasing, and therefore the percentage of legumes in the samples was decreasing. And I know I said I wasn't going to talk about any plot information, but I do want to mention that the additional plot trial that we ran also showed similar effects, where there was an increase in forage production with nitrogen rate, and there was also a increase in the amount of forage biomass in the amount of grass biomass, but there was no significant difference in the amount of legume composition or in the amount of legume biomass in our plots. So there are a number of additional pieces of data analysis that we still have yet to do for this trial. One of them is looking at forage growth rates to see if there is a change in growth that is, to see if a change in forage growth is also measurable with our change in forage biomass within our different rotations. We also have a number of environmental metrics to look at. Volatilization is the loss of nitrogen off of a pasture into the air or off of a field into the air. And we wanted to see how much of the nitrogen that we applied actually did stay on the pasture. When you apply nitrogen to fields, to crop fields, typically it's best practice to put that nitrogen under the soil in some way so that the nitrogen isn't lost. But with a pasture, you are just surface applying this nitrogen. It is a polymer-coated product, so that should prevent a lot of that volatilization, but we wanted to see whether or not that was the case. The other things we're looking at are carbon sequestration and root biomass. So we wanted to see if nitrogen fertility was associated with a change in either carbon in the ground or a change in the amount of root biomass that the plants were producing. We also were interested, and we are interested in looking at the biological fixation by these legumes to see what the legumes are doing and how they're using this nitrogen. Are they using the nitrogen? Are they using their own nitrogen that the bacteria that they are symbiotically related to are giving them? Or are we seeing a change depending on what nitrogen treatment we are looking at? Now, I don't have root biomass data for you tonight, but I do have some root biomass data for you tonight. I don't have nitrogen root biomass data for you tonight. I have been working on this nitrogen root biomass data, and I've been working on the root biomass data that we have collected. We took cores from the pastures and we had separated these cores into different sections. So we had a 0 to 5 centimeter depth, we had a 5 to 15 centimeter depth, and we had a 15 to 30 centimeter depth. And we washed all these roots by depth. One thing that we found, which is very interesting and is a great topic of conversation for pastures and pasture dynamics, is we found a growth in fine roots at the very bottom of the cores in our days post graze. So after grazing, we had sampled, and it took us a while to sample. We couldn't take all of the samples for all of the root biomass cores that we needed in just 1 or 2 days. It took about a week to do all of that sampling. And so over that time, the pastures were presumably they were growing and recovering. And we can see that growth and recovery in the fine roots at the bottom of these cores, which is quite interesting and something that's not seen in literature in pastures specifically. This is an interesting phenomenon because it helps demonstrate that our root data could potentially be useful because we're able to see this fine root growth and we're able to measure it. Oftentimes, root biomass data has a lot of error in it and it is very difficult to see trends, but this is good news. The other thing is that it sort of starts the discussion or adds to the discussion about root sloughing or dieback after grazing. So after plants are grazed, those roots are they sort of they die as well. So the plant is grazed and the plant also loses root biomass. Now, there is a lag time in that regrowth above ground because the plants have to regrow their roots before they start regrowing their above ground plant parts. So, what we're seeing here in the first 12 days after grazing is an increase in that root growth as the roots are recovering before we actually would see much recovery of above ground biomass. And this also speaks to the usefulness of grazing on the landscape because the roots that are dying back in the soil are contributing carbon to the soil. So, grazing your pastures is going to be placing carbon into the soil in a potential way that it could be sequestered into carbon that is ultimately stable. So that is just sort of an interesting data bite that we found with this trial, and we hope to find more root biomass related information. So to conclude and to come back to our nitrogen fertilizer and our goals of this project, this particular range of nitrogen fertilizer, which is a bit low, our top treatment was 90 kilograms per hectare. It has the potential to significantly increase forage biomass while not affecting legume persistence, so legume amount of biomass over time in a pasture. And I just wanted to take a minute to thank everyone who was involved in this project. Totally not just me. I have my very lovely advisor, Kim Schneider, and my co-advisor, Eric Lyons, along with the rest of my advisory committee. Also had some wonderful summer students, Basil and Amelia. The technicians that I worked with, Donna Hancock and Tyson, and all the rest of the forage and service crop students in lab have been very supportive, as well as our funders, the Ontario Agri-Food Alliance and the Beef Farmers of Ontario. Thank you. I'm happy to answer any questions.
Thank you so much, Caroline. Um, yeah, there's a lot of data there. You've got your work cut out for you just getting through all of that. So thank you for sharing your results so far. Um, we do have questions coming in, and I would encourage our audience, please type your questions in the chat as we're talking as well, and I'm happy to field those for you. So just to kind of recap, because we all know researchers do strange things in the name of statistics. How did the grazing management in your project differ from what farmers might typically do? Just so that we can kind of reframe this as what was done because of statistics and how that differs from what we would normally see on a farm.
Yeah, so I guess I have the advantage of working on an additional project with Christine, where we're looking at rotational grazed pastures in Ontario, and the rotation, like the rest period, would be quite a bit longer. You might expect a rest period of about 21 days in May, but that's going to very quickly increase later in the year to be, 45 days, 50 days, depending on the type of commodity you are grazing on pasture. Obviously, sheep have different considerations than beef production. So you're looking at maybe maximum 4 rotations on a typical Ontario pasture, whereas on these pastures, 7 is a lot for Ontario. Just statistically and experimental design wise, we only had 8 paddocks for them to go anywhere. And with the nitrogen effect, because a lot of nitrogen that animals eat also gets excreted, we wanted to make sure that the nitrogen that was in the field that we were treating stayed in the field that we were treating. So we didn't want to move those animals to other places where they could potentially release that nitrogen effect somewhere else. We didn't want that. We wanted to keep them on the 8 fields that we had, or the 8 paddocks that we had within our 8 fields.
Okay, thank you. Um, and actually, just because you touched on this, there was a question just wanting to confirm how long were the rest periods throughout your study?
So the rest periods throughout my study were 24 days.
24 days. Okay, so pretty short once you get out of the spring flush.
Yeah, pretty short. Yeah.
Yeah. Okay. Um, Did
Yeah.
the increase in stocking rate as your nitrogen rate increased, did did that stocking rate increase cover the cost of the nitrogen? Do we know that?
Yes, we do know that. This was one thing that I did actually want to present tonight, but the math wasn't quite convincing enough for me, and I think that might partially be I was using the AgriCorp market values. The price of a cow or the price of a calf has really increased, and so I felt like I needed to check my math before coming out and telling you like how much money potentially could there be profit off of this. But no matter how you add it up, the nitrogen cost is lower than the profit that you would make. So you would have a net profit on those pastures. How much depends on the year, depends on how many animals you're able to stock on those pastures. And I will also throw in the caveat of in 2024, we had almost the same stocking rate on the 30 kilogram per hectare than we did on the zero. And because the zero didn't have a nitrogen cost, but the 30 kilogram per hectare did, that ended up being a loss in profit. But that was just on the 30 and just in the 1 year. The 60 and the 90, they are yielding extra nitrogen, or they're yielding a nitrogen benefit, right? They're yielding a net profit. I can tell you the cost of application per hectare if you want to do your own math with your own animals.
I
Um,
mean,
but
OMAFRA also has field crop budgets. It's publication 60, I think. Field crop budgets. I'm hoping that maybe Rebecca can find the link to that and put it in the chat. You can download it for free. And it gives you some, like just line by line, what would go in a budget for hay. But if you were looking at comparing nitrogen costs, it's the same kind of thing. Your application cost is the same equipment, probably. So that might be a useful tool to start breaking down on pasture what it would cost to apply
yeah,
inputs.
I will say the application cost was actually the same across all treatments because we applied nitrogen and sulfur at the same time. So the 0 kilogram per hectare nitrogen still received some sulfur and was still driven over by the tractor at the time that we applied the other nitrogen. And the other thing is that this was a polymer-coated urea, so the application or the fertilizer cost was a bit higher. So for the 30 kilogram per hectare, it was $107 and 20 cents kilogram per hectare or per hectare. And then the 60 was $182 per hectare, and the 90 was $257 per hectare.
Okay. And because you were working with cow-calf pairs, I just wanted to clarify something.
Yeah.
You did mention an increase in calf production as you increased your nitrogen rates. Was that
Yes.
just based on the stocking rate, so you could stock more pairs, or were those cows also weaning heavier calves?
That was just based on the fact that there was more animals in each field. Each individual calf gained the same amount of weight, regardless of where they were in the trial, if they were on a 0 or on a 90 kilogram per hectare. And they also um, were already born before they went out onto the pasture. So the cow-calf pairs were already established, and we balanced them for heavier calves, lighter calves when they went out, and then monitored how much they gained over time.
Okay. Given how fast Ontario pastures grow in the early spring, so during that first rotation, do you think it might be beneficial to wait until after that first grazing event before applying the slow release nitrogen?
Yes. I don't have any data to make that declaration, though. Um, the one thing about our trial is that the fencing at the Elora research station was not conducive to applying nitrogen after the animals went out on pasture. So we applied the nitrogen at green up because the fences were down. and the tractor could go in and apply that nitrogen and come out. And there wouldn't be any animals in there. We would have had to move the animals somewhere else or apply the nitrogen as the animals were going around. Um, or it would have taken us 24 days to apply all that nitrogen to each paddock individually. And the tractors don't really quite fit on into the fields that way.
It's hard to turn them around on just 1 acre. Yeah.
It's hard to turn a tractor around on just 1 acre. I've done it. It's hard, especially with a big you know, fertilizer distributor on the back as well.
Got it. So with your plot work, did you do anything to determine which grass species in the mix had the greatest response to nitrogen and which grasses had the poorest response to nitrogen?
Um, so the I kind of maybe buried the explanation there for the split by plot trial, or the split in our plot trial. We had just a grass mix on our plot trial, so all the grasses combined. We had split out our legumes because we were more interested in the legume effect, or the nitrogen effect on the legumes and not necessarily on the grasses. However, I do know from literature, so this is not my research, it's someone else's research. Orchard grass tends to have a higher nitrogen response than the other grasses on my trial, the tall fescue, meadow fescue, perennial ryegrass, um, and Kentucky bluegrass. Perennial ryegrass is probably also close with the response, but orchard grass tends to have a higher response just because it can yield a larger plant overall than perennial ryegrass. So that would
Okay.
be what I would expect if I had done that testing.
hmm
Yeah.
And when you did the quality analysis, did you see any differences in the fiber content of the forages? So NDF or ADF between your treatments?
No, no, there were no differences between those treatments. Um, May and August would have differences, though, with there being more fiber in August than there was in May.
Okay. And how were phosphorus and potassium managed? So did soil levels decline more for those nutrients in the higher nitrogen treatments?
I would have to look back at our soil tests to give you a definitive answer, but I know how we managed P and K is we sampled the sections of our fields, the 1 acre sections, and then submitted those to be soil tested. And then we applied the rates of P and K that were um, recommended in the forage production guidelines. Um, so we managed based on like on an as needed basis. Um,
From this book.
from that book.
Okay.
Yes. Yes. Oh, there it's actually okay. There are two places you can find the same fertility guidelines, same tables. And I was kind of and my brain was fusing them both together. Um, so from what I can recall, it was more so a landscape effect as to which fields required P and K more all across the 3 years than it was a nitrogen effect. We did have one side of our fields that were a little bit wetter than the other side of our whole trial. And there was a bit of a slope as well. So that created the variability in the differences in P and K applications. I didn't notice a nitrogen effect on P and K. But it is an interesting question. I'm gonna look back on my
And
tests now.
yeah, and did you only apply P and K before the trial started, or was there an application every year?
So there we tested and applied every year in the fall.
Okay.
Yeah.
Were you using the same pasture fields every year? And did each field get the same rate of nitrogen each year?
Yes and yes. Yeah.
Okay.
So it was a potentially a cumulative effect of the nitrogen over time. So the 0 kilogram per hectare nitrogen treatment received 0 kilogram per hectare of nitrogen every year. So it didn't receive any nitrogen fertility at all, except for the urine or manure from the animals.
Okay. And you mentioned that all of the treatments, even the zero nitrogen treatment, got some sulfur. So what sulfur rates were you using?
10 kilograms per hectare.
Okay, so about 9 pounds an acre. All right.
Great. I love I have no ability to convert to imperial.
I have done the metric to imperial enough that I know kilograms per hectare to pounds per acre is times zero point 0.9 and 10 is an easy one to convert. Yeah. Um, all right, I think we are caught up on questions, but I've got another question for you. Oh, see, I said it because Chris snuck one in here. How many days were the cattle in a paddock before being moved? So how long were your graze periods?
So either 2 or 3 days, depending on how much forage there was. Yeah.
Okay. And so the goal was to graze down to that a specific target residual. Is that right?
Yes, the goal was to graze down to a specific target residual. We used a tool called a rising plate meter to measure our pastures and measure that residual. But I have done some eyeballing and some thinking with rulers and some rising plate meter stuff. It was about 15, just under 15 centimeters, that we were targeting as a residual, which is just under 6 inches. I did convert that one to
Well,
into
that
imperial.
yeah. So what was something that you learned from this trial that surprised you?
Um, Okay, I have two things. One thing, when the trial was happening, and when it started in 2023, um, I I had no pasture management experience, and obviously I was not the pasture manager. I had some wonderful expertise from people who were telling me what to do, when to move the animals, what to look for. Um, so I guess in that sense, a lot of things surprised me about pasture management, how you I look at a pasture to see how much forage there might be or when the animals are ready to move. Um, and in that sense, what surprised me was truly how fast the grass was growing in the spring, or how fast that forage biomass is growing in the spring. We had some challenges with that because the pasture can grow faster than it gets eaten in the spring. And so that becomes a research challenge and also becomes surprising because although I did say 24 days was our length of rest between rotations or our length of rotation, we did have some times in the spring where we came back after 16 days to those top pastures because we just needed to keep that forage biomass at the increments that we were targeting. And we had to move them back because that was our only strategy of keeping the forage biomass at the level that we wanted. We didn't really have the option to take anything for hay.
All right. So given that you had more grass when you applied more nitrogen, do you think that you could have grazed tighter, so taken it down to a lower residual without any negative impact on the grass plants?
Um, I think I think that's something I still haven't really become confident with or I'm sure of. I know a lot of the time in um, pasture management in Ontario and also out west, there's the idea of keeping those residuals higher, particularly later in the season. Um, we did move our stocking rates around in the spring to try to hit those higher nitrogens harder, because we had underestimated the amount that those plants would be growing and how much the animals needed to eat. So we actually did start like moving animals and like adding more animals in to those higher nitrogen treatments to get that residual lower. What I would have liked in the treatment was obviously to know in advance how many animals could be on there. But with the weather, you can't really know. And we had a dry spring in 2023, so we weren't quite sure what a regular wet spring in 2024 would end up being like.
Hmm. Oh, just when I think I get to ask my final question, they drop more good ones in the chat. On this project, did you have any concerns about nitrate poisoning, particularly on the high N rate treatment?
Um, we I did talk to some specialists because typically rates of nitrogen applied in one sitting are 75 kilograms per hectare. It's not recommended that you apply more than that in one application. But um, in this case, because it was that slow release nitrogen over that 60 days, it was determined that we could put that nitrogen application on in a single application. We did do some nitrate forage quality testing and didn't see anything that would suggest that there would be nitrogen or nitrate poisoning concerns. So.
Okay. What was your criteria for adding or taking away animals in terms of adjusting that stocking rate?
So in 2023, we started with all the treatments having the same number of animals because we weren't quite sure what the nitrogen effect would be. And we had decided to remove the animals. We decided that we wanted to graze each of the nitrogen treatments to the same residual. So if we were not hitting that residual target, we were adding more cows or we were removing cows. So that was the criteria that we used. And um, we did have a acceptable entrance putting in value for the zero nitrogen treatment. Obviously, we couldn't have the um, putting in be the same for like across all the nitrogen treatments because we had more biomass produced on the higher nitrogen treatment, so those were higher. We did have like a minimum for 0 kilogram per hectare, but I did not convert that from a rising plate meter value into a regular non-compressed forage value. So I wouldn't want to take a guess.
Fair enough. Um, were the calves fed any supplemental feed, or did they just have grass and mama's milk?
Grass, mama's milk, mineral, that there was no supplemental feed on the pasture for the reason that I had mentioned before is that we wanted the nitrogen to stay in the system and we didn't want to add anything in or remove anything in once those fields were put together.
Okay. What was the grass to legume ratio in the pasture mix? I'm gonna assume this was at seeding because it changes depending on how it catches.
Yes, um but when I got there in 2023, it was about between 20 and 25% legume, dry
Okay.
matter.
And sorry, was that the establishment year or was that already into a production year?
That was the first production year.
Okay.
Yeah.
Um, long term. Oh, this is not a question, this is a comment. Uh, John thinks long term with increased biomass production from N, the cost of P and K replacement needs to be considered from a profitability standpoint. That's a great point, John. A higher yielding crop is going to have greater removal values of other nutrients too. So that's a great point to bring up.
Yeah, I appreciate that point. It's not something I had really had on my radar, so I will be pulling those tests that we have to add that to the discussion that I'm developing. Thank you.
All right. Last question. If there's one thing that you want producers listening to tonight's webinar to take from your findings so far, what would it be?
I think one thing important to know or important to think about is oftentimes in Ontario, we don't think about pastures as being something we put a lot of inputs on or a lot of nitrogen on. And I think this trial demonstrates that adding nitrogen fertility to pastures is going to give potentially an increase in forage yield. And if you can manage that forage yield, that increased forage yield, then it might be something to consider.
All
If you
right.
can manage it though. If you're not prepared to add more animals and use it, then there's no point. Yeah.
Okay. Well, thank you so much, Caroline. I'm going to turn things back to Joe.
Thanks.
Okay. Thank you so much, Caroline. This was excellent and obviously struck a nerve because there were some amazing questions in the chat and a really good discussion after the presentation as well. Um, First off, I'd like to take a moment and thank our sponsors again, Trillium Mutual Insurance, Brussels Agri Service, Range Ward, and the Beef Farmers of Ontario. Thank you.